All right, we'll go ahead and get started. Thank you all for coming. Today it's my pleasure to introduce Dr. Tina Del Carpio from my lab. Tina just finished with their PhD in EEB this past winter quarter. And so now Tina's back to tell us about some of their work in the lab. So first, just a little bit about Tina. Tina came to UCLA with an NSF GRFP to work in the lab of Bob Wayne on a number of projects related to canid genomics. And so after several years in Bob's lab, Tina transferred over to my lab, first in a co-advising situation, and then over to my lab. And well, Tina will tell you more specifically about the specifics of their projects and their research. But what's been really, I think, special about Tina was how they crafted their own path and were quite independent with their projects, some of which were not really in line with everything that we had going in the lab. And so it's a testament to Tina to pull off that work. Throughout Tina's time at UCLA, they've been involved in a number of projects and efforts related to EDI leadership at the department level and at the university level. And Tina's contributions there have also been exceptional as well. And so now Tina is a data analyst at the California Interagency Council on Homelessness. They just started this position last month, and so we'll be putting some of the bioinformatics skills that Tina learned here to good use. So without further ado, I'll turn things over to Tina.
Yeah, thanks for that introduction, Kirk. Yeah, so my talk today is going to be just a sort of more of a story about my dissertation journey, how I went from doing ecology work, and like Kirk said, now I'm working on homelessness data for the state of California. And so I want to sort of, as those of you in the department know, we don't have a formal defense. So this is just sort of a nice closing talk to, reflect upon my journey here at UCLA. Before I get into things, I want to start with a land acknowledgement. The UCLA community acknowledges our presence on the traditional, ancestral, and unceded territory of the Gabrielino/Tongva peoples, and I acknowledge the use of samples collected on the lands of the ancestral and modern Inuit and Inuvialuit peoples. I also have to say, as a government employee, that the opinions that I am expressing are my own and not those of the California Interagency Council on Homelessness. Certainly to the students in the crowd who are thinking about what you want to do after grad school, if you want to learn about at least how to apply and get a government job in the state, happy to talk about that. And I'll talk a little bit about my work there at the end. But yeah, so I wanted to take you on this journey of how I went from actually studying animal behavior at Duke to completing my PhD in biology in Kirk's lab, and to now working at Cal ICH. So after undergrad, I spent 5 years teaching molecular biology, and I also taught a general evolution class, mostly for freshmen and sophomores. And so I thought a lot about, introducing folks to thinking about evolution and natural selection on sort of a deeper level. And I found that while teaching and also while doing volunteer work, working with general audiences, that a fundamental concept that sometimes gets overlooked, even though we think about it in the beginning, but can get overlooked, is the fact that evolution is not goal-oriented. Organisms are not trying to do something that will be advantageous for them. Rather, natural acts on the natural variation that exists. And so to sort of think about that, I'll bring us, all the biologists in the room familiar with this classic example of the peppered moth. So There's these moths in Europe where they have two different forms. They have this lighter form and this darker form. And before the Industrial Revolution, most of the moths were this lighter color. And then post-Industrial Revolution, particularly in urban areas, most of the moths ended up being this black color. And the reason for that is the change in the environment, where in this unpolluted environment, you had trees that had lichen growing on them. And as you can see, here's the light form of the moth, blends in very well with the lichen, whereas the black form sticks out a little bit. But then in this polluted environment, post-Industrial Revolution, the lichen died off, and so more of the tree bark was exposed, and these black moths blended in better. So there was selection towards this black form or morph in the urban environments. But, you know, something to think about is, you know, it wasn't that the moths were purposely, you know, turning darker colors once the environment changed. Rather, that black morph or that black form already existed. And we can think about, but what if all the peppered moths were these light colors? So, you know, we can't know for sure what would have happened, but we know that it would definitely be a different story if this natural variation of the black morph didn't exist. Yeah, and so thinking about how this natural variation is required for adaptation has always been an interest of mine, and driving my interest in thinking about biological diversity. And then specifically with my background teaching molecular biology, and I also worked as a lab manager doing studying genetics of baboons with Jenny Tung, I came to UCLA particularly interested in genetic diversity. And then while I was here at UCLA working with Kirk, I started thinking about projects looking at this molecular process called recombination. So this is a process that happens when eggs or sperm are being produced and DNA gets shuffled around. So these X's here, we have our DNA. This is from parent 1 and parent 2. And you can imagine they have different DNA sequences or they have different alleles. And during this process where recombination happens, this is the actual like swapping of genetic material between these two lines of chromosomes. And then you end up with these chromosomes that have a mixture of DNA sequences. So they have the alleles or the sequences from the blue chromosome, and then here from the red chromosome. And then here you also have the mixture of the red and the blue. So recombination contributes to genetic variation by swapping around what DNA sequences or alleles are inherited together. And something that I found interesting about recombination is that where and how often recombination happens itself can vary. So there's variation on the levels of genomes. So along the chromosome, there's differences between individuals, sexes, populations, and species. And then there's also this interesting work looking at closely related species. So one example specifically in humans and chimps found this surprising result that hotspots, or these areas where recombination is concentrated, can differ between humans and chimps at fine scale, or very like focused, looking at the locations of these hotspots. And then another piece that came into our work, like Kirk said, I came in to UCLA working with Bob Wayne, who has a history of programs working with canid, so wolves and dogs. And so we're really interested in domestication. And there's this theory that domestication increases recombination rates, partially, you know, coming from the idea that variation is a key for the domestication process, because it can create different combinations of these traits. But most studies have tested this theory mostly in plants, and those results have been sort of mixed. Some have followed this trend of increased recombination and others haven't. But there's been a lot fewer studies in mammals. So coming back to thinking about dogs and wolves, of course, a great model for domestication. Dogs are thought to be the oldest domesticated species. And then one study that did look at recombination in mammals did not find this increased recombination trait in dogs and wolves, but their search was limited to 20 genes in the genome, so only a small portion of the genome. Also, previous studies have looked at recombination rates, but solely in mixed breed dogs. So there's an opportunity here to look at more dog breeds and to also consider wolves as a comparison to look at this domestication animal. And so for my first project, I was thinking about, are there differences in broad scale recombination rates between dogs and wolves? So has, domestication potentially altered these patterns of recombination? And so I estimated recombination rates in wolves and 4 dog breeds. So like the prior study I mentioned before focused on 20 gene regions. I focused on whole genome sequences that were high coverage, so high quality DNA sequences from one population of North American gray wolves in Canada, also border collies, Labrador retrievers, Tibetan mastiffs, and pugs. And so I estimated the recombination based on shared patterns of genetic variation within each group. So I showed you that, let's see, I can go back. This image here about how recombination can, create these patterns of like the red and blue chromosome. We looked at those patterns of how the chromosomes had shared pieces of chromosome were inherited. So we use that genetic variation to estimate the recombination. And what I found is that estimates of recombination does vary between some dog breeds and wolves. So I'll walk you through how to read this plot. So here on the y-axis, I have a measure of the recombination rate. And then here on the y-axis, and then on the x-axis, here I have the different populations. So we have wolves, border collies, labs, pugs, and Tibetan mastiffs. And what I did was I took the recombination rate of each chromosome. So there's 38 chromosomes in canids. And so I plotted the range of recombination rates chromosome wide. And then looking at this is a box and whisker plot. This sort of shows you the distribution of the percentiles of like, here's 25% of the data is here, 25% here in the top half, and then here as well. So you can kind of focus on here, this line in the middle is the mean. And so if we just compare the means between wolves and then these 4 dog breeds, you can see that the wolf pattern looks very similar to labs and Tibetan mastiffs. But in border collies, we have this really elevated recombination rate, like we have some points up here even. And then in pugs, I saw this really decreased recombination rate. So I found that some dog breeds do have recombination rates that vary from wolves, but I didn't find this pattern that's theorized that recombination rates would be increased in domesticated species. And so this was kind of a surprising result to me, at least. I came into this project expecting to find fewer differences. And so I was sharing this work and thinking with others about how to write up the paper on that work. And one person I reached out to was Omar Cornejo, who's currently at UC Santa Cruz. And when I shared this work with Omar, he brought a question to me about how does recombination rate impact the estimates of the distribution of fitness? And I'll talk in a moment about what the distribution of fitness, or DFE, is. But so together, Omar and I were able to think about, how do some of his interests align with my interests in the data that I already had available, that I had created from the wolf DNA sequences. So thinking about what the DFE is, the DFE describes how new mutations impact the fitness of an organism. And because this sort of looks at, when these mutations arise, whether how deleterious or, negative or not they'll be for the organism, tells you about how selection acts on these new mutations, and in turn, how that selection shapes genetic variation. So this project for me sort of came back to thinking about this idea of diversity and genetic variation. And part of the reason Omar had this question about why recombination might impact our estimates of the DFE is because to model and infer the DFE, we have to make assumptions about the recombination rate. Basically, many models assume that there's free recombination and that sites are unlinked. And part of the reason we build models with assumptions like this is to make the calculations feasible either in computational time or sort of memory resources. But we also need to consider how do violations of this assumption affect the results. So Omar and I came together to think about, does recombination rate influence estimates of the DFE in wolves? And so what I did for this project is I divided the wolf genome based on recombination rates. And then for those different recombination rates, I estimated the DFE. So I broke up the genome into 1 megabase or million bases. And then for each of those sections of the genome, I estimated the recombination rate. So using the results I had from my prior project. And then I labeled those recombination rates into 3 groups of a low, medium, and high recombination rate, basically dividing the rates into 3 quartiles. And then, for those 3 groups and for the whole wolf genome, I estimated the DFE. And what I found is that the DFE in wolves varies somewhat with recombination rate. So let me walk you through how to read this plot. So here on the x-axis, we have the selection effect, which this is basically how new mutations are expected to impact the fitness of the organism. And we focused mostly on neutral and then deleterious variation, because from the literature, we know that most new mutations fall into these categories. So you can think about, so these are nearly neutral, so mutations that would have little to no effect on the organism. And then over here, we have mutations that would be strongly deleterious or negative to the organism. And then here on the y-axis, we have density, or you can think about this as a proportion. And so we're showing how many of new mutations are neutral versus these different levels of deleterious. And then I have 4 different colors here. So these white bars represent the whole genome or all recombination rates. And then I have in blue my low recombination rates, medium, and then high recombination rates. And so my question was, are these patterns going to vary based on recombination rate? And you can see looking at these bar plots that this purple one seems to be very different from the other ones. The low, medium, and all recombination rates are all very similar to each other in all of these categories, but the medium recombination rate is skewed towards this strongly deleterious variation. So I found that the DFE varies somewhat with recombination rate in wolves, but then we were thinking about, what is sort of the biological impact of these differences. And to contextualize that, we thought about the DFE from other organisms. So this plot is set up the same way, where we have the selection effect here, and then the density on the y-axis. And this is a modified plot from a review that Chris did in Kirk's lab, where Chris looked at DFE estimates in a wide range of species that were available. And you can see they're color-coded here, but I want to highlight looking at the wolves. I added our medium recombination rate in the sort of salmon color, or sorry, in the orange color is the medium recombination rate. And then in red, we have the all recombination rate. And so, you know, to the lab, having looked at the DFEs of these other organisms, it was very striking how skewed towards this strongly deleterious variation the wolves are. And so you can think about this of, even though this medium recombination rate bin looks different from the genome-wide wolf recombination, or yeah, the genome-wide wolf DFE, if you looked at these different patterns and especially focus on the strongly deleterious variation, you find that the medium recombination rate for wolves looks much more similar to the all recombination rate compared to these other organisms. So in summary, even though the DFE for wolves did vary somewhat with the recombination rate, when you look at it compared to other organisms, you wouldn't mistake it for something other than a wolf. I will take a quick break because I forgot to plug in my computer. So a little 30-second intermission. Right. Okay. Crisis averted. Much better than my Apple Pencil. One of my major complaints is the Apple Pencil doesn't warn you that it has low battery until it's at 5%. And you're like in the middle of a meeting and you're like, there's no way to change it as far as I know. Be like, could you give me a heads up at like 25%? So, all right. And so now we're gonna shift away from thinking about some of our more evolutionary focused projects, because as I got into the later stages of my PhD, you know, like all PhD students, I was evaluating, what do I want to do with my PhD? You know, sort of being in academia longer and seeing some of the options. And someone who really changed that path for me was Diana Azurdia, Dr. Diana Azurdia, who's the director of recruitment and inclusion for the bioscience research training program here at UCLA. And so working with Diana through like SACNAS, the Society for the Advancement of Chicanos, Hispanics, and Native Americans, and other projects, I started thinking about diversity at the level of graduate school programs, which was something that was already kind of interest to me as like the child of a Venezuelan immigrant and a trans and queer person, thinking about, you know, who are the people who make up academia, and how, you know, one of the filters to get it, you know, to continue along this path is succeeding through graduate school. And so thinking about that, Diana and also one of my committee members, Paul Barber, connected me with this program, Competitive Edge. It's a UCLA program that's meant to support a more diverse PhD student body. And what it is it's a 6-week intensive, what they call bridge program. So sort of during a transition period in your career for science, technology, engineering, math, or STEM PhD students from historically excluded and underrepresented groups, or URG students. And the program has a lot of components that are focused on professional development. So for example, there's training on how to be a mentee and how to work with your advisor, which is a central part of the STEM PhD experience. There's also various workshops on research-related skills, such as coding in Tableau, and also, you know, how to do a literature review, things like that. There are also, you know, opportunities for socialization, so connecting with a cohort of fellow historically excluded and underrepresented students, and then also past cohorts and also faculty members from these groups. Program also includes financial support in the form of a stipend. And during this time, too, students work on a research project with their potential PhD advisor. So they have an opportunity to get started on some mentored research. And so the question I was interested in about the Competitive Edge program was describing how does the program benefit students. And to assess this, I surveyed Competitive Edge students. I had 14 students respond about their experiences out of about 40, which is a typical survey response rate. And these were all first-year STEM PhD students who participated in the program in the summer of 2021. And I surveyed them at the end of their first year, so in June 2022, so they would have a chance to reflect on their first year in graduate school and think about how their participation in the program influenced their acclimation, their start at graduate school. And so one of the main questions I asked them, I'm going to focus on, is I asked these students directly if the Competitive Edge program improved their experiences in these 5 areas. So I looked at research skills, sense of belonging, self-confidence, overall well-being, and interactions with their advisor. And the reason I picked these 5 areas is because they relate to success as a graduate student. So looking at the literature about the reasons that graduate students leave their programs versus students who stay and complete their intended degree. These were all areas that affected that. And also there were areas that were targeted by the Competitive Edge program. There were components I expected the program would help students. And so looking at the results, I found that Competitive Ed does improve student experiences in these 5 categories. So here on the x-axis, I have those 5 different areas I was considering. And then on the y-axis here, I have agreement. So the questions were formed as a Likert scale question, which you've probably seen before, of, mark how much you agree with this statement. And the statement of the Competitive Edge program improved my, and then research skills, sense of belonging, so on and so forth. So you can see, down here is strongly disagree, and then up here strongly agree. And again, I have these sort of box and whisker plots. Like I showed you before, there's little dots here that each represent a student's response. And these black dots here represent the mean. So I would focus looking at where these mean or these black dots fall. And you can see for all of these scales, the mean falls somewhere in the agree category. And as we go to the right here, we have higher means. So like especially interactions with my advisor, all the students agreed. Half of whom strongly agreed that the program improved their experiences in those areas. So the students, found that it did improve their experiences. Then I also, thinking about describing it, I wanted to go beyond just quantitative or sort of numerically quantifying their experiences. And I also wanted to get into just sort of more qualitative or descriptors of their experiences. And so I asked students to describe how the program impacted their first year. And I want to share with you some of the quotes that I think really summarized what the students said. So one student commented, CE did not solely focus on preparing us to conduct research, but focused on other aspects that are necessary yet forgotten, such as building self-efficacy and setting expectations with a new mentor. Also, I really appreciated the opportunity to do a small research project and start working with my advisor before the beginning of the school year. And then I thought very impactful, one of the students said, because of Competitive Edge, I felt like I belonged at UCLA. So in addition to, agreeing with statements that the program improve their acclimation during their first year of graduate school. They also put into words why that helped. And so I think this really captured that many of the responses talked about how they liked that there were workshops and components that were not just about technical research skills, but also about sort of navigating these other parts of graduate school, like working with your advisor, this building self-efficacy sort of builds into like belief in yourself that you can succeed. And then students also really appreciated that this was an opportunity to get started with working with their advisor early on. So when you start graduate school, often you're taking classes and you're sort of being, learning the lay of the land and getting pulled in different directions. But because this program, students had to have an advisor who was dedicated to working with them during the 6 weeks, they had a lot of hands-on time working with their advisors to get started on their research projects. And, like all of the academics in the rooms know, often your early research projects in graduate school don't make it into your dissertation. And so this was an opportunity for students to really start testing the waters and testing, what projects they want to work on and, have an opportunity to do that. And then, the sense of belonging is a really important component of this program, I think, and especially from my own experiences and from looking at the literature, especially for students from historically excluded and underrepresented groups, having some kind of social network and support in navigating the nuances, or it's often called the hidden curriculum of academia, is really important. And so, having this sense that literally you belong here at UCLA because of this program, I think it was a really important comment. Yeah, and then I'm gonna wrap up my talk just introducing a little bit of what the work that I'm doing now. So after my work with Competitive Edge, I became really interested in a career path that was sort of more on this social science end of academia, of looking at the experiences of human populations instead of canid populations, and using my data skills to wrangle and understand this data. And so when I was looking for work that was in this data analyst sort of sphere, but I felt was doing a social good, I sort of looked a lot at nonprofits and government agencies as well. Seemed to be where a lot of that work was concentrated. And where I landed is Cal ICH, or the Interagency Council on Homelessness. And, I'm sure everybody is aware that this is homelessness is a major issue, particularly in California. But just to share some of the numbers on this, once a year in January, volunteers and staff members go out and count all of the people experiencing what's called unsheltered homelessness. So people who are living either just on the street or in structures that are not meant to be habitable. So like condemned buildings or like cars. And so staff and volunteers across the country go out and count as many individuals as they're able to find to better understand, the scope of homelessness. And then also there's data about individuals who are in what's called sheltered homelessness, or in like a day shelter or an emergency shelter, or there's also transitional housing programs. And so this report that was just released about the count in 2023 found that across the US, more than 650,000 people were experiencing homelessness. And then more than 181,000 of those people were specifically in California. And then getting even more granular, thinking about Los Angeles with this Los Angeles City and some of the surrounding areas in the county, was the region with the second most people experiencing homelessness, with over 70,000 people. So, of course, the data backs up our experience of, I know as we all go out, we see our unhoused neighbors and we see the problem that exists. And while, some of this data can be depressing and sort of a hard note to end on, collecting and analyzing this data on homelessness informs our action plan for preventing and ending homelessness in California. So, we collect and look at data, not just, how many people are experiencing homelessness, but we also look at, for example, racial and ethnic disparities that happen, like Black and African American populations in California experience homelessness at a much higher rate than they make up of the population in California. We're also doing work where currently our estimates of Native or Indigenous individuals who are experiencing homelessness in California are undercounted because of a lack of interactions with tribal communities. And so that's work that Cal ICH is also doing. Yeah, and so we're working on, understanding who's experiencing homelessness, what are the barriers they're experiencing, what are their trajectories through the system. So using services like temporary housing or programs that are called like rapid rehousing in California, so that, hopefully in our lifetime, we can move towards preventing and ending homelessness in California. Yeah, and on that, I want to end with some thank yous. There are so many people to thank, but so I pulled up some people I'd like to thank, especially all of my committee members. Of like Kirk said, and as you saw, I went on this non-traditional path, and so I really appreciate that Kirk and the rest of my committee supported me in including this more social science chapter of my dissertation. I had a lot of support from my cohort mates, so the people who started the program with me. Especially I want to shout out Liz, Evan, and Christian. Also, so many people in the Lohmuller and Wayne labs supported me. Jesse, John, Maria, and Pedro, and Eduardo helped me so much with all of the coding and actual details of the research I did in the lab. And Annabelle, Maishi, and Chris also provided some technical support, but I think also more emotional and social support through my graduate program. I also have academic collaborators and friends, so Omar working with me on that DFE project, and Tani and Rachel that I worked with at Duke who helped me make the transition to UCLA. There's also, of course, so many people at UCLA, but I especially want to shout out Jenny and Eric, who run the genomic analysis training program, Tessa and Morgan, also within EEB, provided also a lot of this emotional and social support through my program. And then, of course, friends and family, my mom who flew in from the East Coast to be here in person for my talk. One of my best friends, Lawrence, who I talked with every week about my dissertation and his dissertation, and now we're both doctors. And then, of course, my fiance, Tess, and my cats, Tuka and Jem. And then, of course, my funding sources at NSF and at the NHGRI. Yeah, and then, of course, here's Tuka and Jem. Yeah, and so I want to thank all of you, this packed room of people who came to see my talk, and all the people on Zoom. And so with that, I'll turn it over to some questions. Yeah, Mark.
So,
Thank you.
The stuff where you looked at the recombinations.
Yeah.
Like dogs, so it looked like bugs are like super low. So I was curious, is it just because they have like is the data set really like diversity, or I know like they have really bad, like their bones are really bad.
Yeah, yeah.
So like I'm curious what's going on.
Yeah, certainly this is a question everybody has asked, even like, Mark has worked more on the sort of the paleo side with canids in Blair's lab. But also even just lay people are like, when you look at a pug, it's pretty weird, right? Like they can't really breathe so good, things like that. We're not entirely sure how that might relate. I actually spent some time trying to talk to a dog breeder who like has a history of like breeding pugs. I mean, I did learn a little bit about some of this I knew already, that, dog breeding is really intense. And like in the case of like show dogs and things, they actually will freeze semen. And so a male individual can continue siring youth right well after they have died even. Like these breeders will have like develop, and they even call it the breed lines. So I think some of it is just this really intense artificial selection that's happening in dogs, but I don't know necessarily how sort of the morphology of pugs relates to that versus just sort of generally there's very distinct things happening in each breed. And, it could just be chance that somewhere along the way, some of these sires from pugs had this low recombination rate. We've also thought a lot too about, maybe there's other things that are biasing and impacting our estimates. So I've done some work in Pedro, especially did a lot of simulations with me looking at differences in population history. And like also we looked at, as you know, Mark, dogs have, in general, all dogs have this decreased genetic diversity. I didn't see specifically that pugs had like significantly less genetic diversity than the other dog breeds, but they did have significantly more or less linkage, disequilibrium. I never remember the direction it goes, even though I've studied recombination. LD, like the patterns of LD, which I used to infer these recombination rates, like we did see pugs had a difference in that pattern that correlates with this difference in recombination. So I think in short, we don't know for sure if it relates to, why are pugs so weird, but maybe just in general, the strangeness of this really intense artificial selection. Yeah.
I was wondering about the theory that you were testing. So what was the reason, theoretical reason for thinking that you would have lower recombination or higher recombination rates in domestic breeds, or that it would be related to?
Yeah. So I think this is, really more just sort of like I guess like most theories, like sort of a thought experiment. And so I think the idea that Gornal had was that variation is important for domestication. And so, right, that you're sort of, you might have traits that you want in whatever you're trying to breed, whether it be agricultural or animals. Right. So there's different traits that you want to select for, and that recombination would give you more patterns of those traits. And so that this increased recombination would give you a better chance of sort of hitting this mix of traits that you want to preserve. But then sort of in our conversations too, like especially I think Paul Barber brought up though, once you hit these combination of traits you want, you're not going to want to break them up. And so there's also sort of this pull that, maybe you don't want increased recombination. You want to, right, keep inheriting these traits together.
So the idea was that there's actually selection for higher recombination rates in domesticated lineages, or that people tend to use lineages that have high recombination rates and are more successful, I guess, with those because there's more variation to work with.
Yeah. Basically, I mean, right, it's sort of that this artificial selection was, right, that the breeders are sort of unintentionally selecting for this higher recombination rate because they're getting, right, this more variation in the traits they want was, or combinations of advantageous traits. Yeah. Yeah, of course. Okay, I will transition to, I know I have some questions in the chat. Oh, somebody asked about this plot. Okay, if I can go back over. Someone asked about this plot again, where we have recombination rate, and then we have wolves, and then the different dog populations. They said, I'm unclear about this graphic as well. Does this mean that wolves are more like labs and Tibetan Mastiffs than the two other breeds? Yes, as far as patterns of recombination go, labs and Tibetan Mastiffs that resemble wolves much more. And then somebody also asked in the chat, did you find it difficult to market your skills as a bioscience PhD student to a social science audience? I think actually it wasn't that hard because, I just focused, I don't know, there's lots of talks about this of being an academic, and if you want to transition to an industry or government or nonprofit track, there's lots of services and people who talk about this, thinking about what's called transferable skills. So basically, what skills do you have you developed that can apply in these different contexts? And so, as a PhD student, you develop a lot of these sort of general problem solving skills and, developing hypotheses and testing them. And especially, I was able to, like Kirk said, flex some of my bioinformatics skills of the fact that I can code in Python, or especially in R. Like in my role, I use R a lot. Also, surprisingly, a lot of Excel, actually. You can do a surprising amount of things, or at least it was surprising to me how many things you can do in Excel using macros and things. So my colleagues are also teaching me about that. But you'd also be surprised how many people, don't really know their way around an Excel sheet. But yeah, I didn't find it that difficult. I just, focused on what are the skills that I have that are just sort of general, like problem solving and data analysis skills. Okay, I have another question looking at this plot. Does this mean there's a wide variation in border collies? Yes. Yeah, as you can tell with all these other populations, these plots are kind of smushed together. And so I had to extend the plot up here to capture some of these higher rates in border collies. And so as you might recall, each of the points I was looking at here is a chromosome. And so some of the chromosomes in border collies have, this 4 × 10⁻⁸ rate, and some have all the way down to like 0.5 × 10⁻⁸. So yeah, there is also this trend where border collies have more variation. Okay. Yeah, Victoria?
2 questions, because about different parts. And
Sure.
Very much.
Thank you.
So one is on the border collies, since that slide is up. Isn't it possible that what people call the border collies could be kind of more mixed between other species? Is it likely, and couldn't that generate that kind of higher value of recombination, higher in border collies as a quote unquote, whatever you call subspecies?
Yeah, this is a breed.
The border collies are comprised of more than one, and that would generate certainly the variants, but maybe even the higher rates.
Yeah, I mean, I think that gets back to what I was talking about in Mark's question of, like, again, this really extreme situation with dogs and dog breeding. Yeah, and it's possible that sort of the lines that the border collie breeders are using are more varied than sort of what the pug folks are using, for example.
So I'll get to the second part. The part I was very interested in competitive edge, because we
Yeah.
always want to see if these programs are achieving the outcomes that we like. It would be interesting, and when I've done these reports for social science, it's so hard to get the kind of control groups that we want when we do biological samples. Two control groups that I could think of is, first of all, the data do show that the people who took it really appreciated it and it made an impact on them. The question is the people who didn't respond, it'd be nice to know if they didn't think it had an effect, in which case this would argue that maybe what you do is open competitive edge to people who don't want it. But it's the other is, let's just say what would be the control group to people who didn't, who didn't get in, and whether or not they would have benefited, like, you probably have thought more about control groups than I'm bringing up. And then I like the fact that you're now showing these other measures of
Ready.
success are better than self-reports. But I don't know if you went out long enough. So can you just comment on this general?
Yeah.
Sense? It's very hard in social science research to match the designs that we like as biologists.
Yeah. This was certainly a thing we thought a lot about, especially me and Diana. And like Diana also has a background in STEM and in bioinformatics sort of work. So we collaborated with a lot of people who do more social science and thought about how to have these control groups. So what I didn't show in my other plot is I did also survey first year students during the same time who did not do the competitive edge program. And then I had two categories within that. I had students who fell into these historically excluded, underrepresented, or URG groups, which is here, the non-competitive edge URG groups. And then I also had the well-represented students. And then I have the CE students. This is also self-reported data. But basically I asked students to rate themselves in 8 different skills. So research, connection to resources, evaluating journal articles, faculty interactions, financial literacy, mental well-being strategies, science communication, and writing fellowship applications. Again, these were skills that we know relate to success as a graduate student, and also are skills or areas that are targeted by the competitive edge program. And so I asked students to rate these at the end of their first year. I asked them to think back to before they started their PhD program, or for the competitive edge students before they started competitive edge, and rate themselves on these 8 skills. And then I asked them to then think about themselves currently. So at the end of their first year, to rate themselves on those skills. And again, this was the Likert scale from 1 to 5. And then I looked at the change in their responses. So, I took their end of year scores minus their first year scores. And I plotted those. Here in white, we have the means. And again, this is the competitive edge group, the non-competitive edge URG group, and then the well-represented group. And so I found this trend that if you look across these, through these different areas, in almost all of them, the competitive edge students had the highest mean. And then I also did do some statistical tests. I just did a t-test comparing competitive edge students to the URG group, and then competitive edge students against the well-represented group. And I did find a statistical significance here with connection to resources between competitive edge and the non-competitive edge URG students, and then also faculty interactions between the competitive edge and the well-represented students. But I will also say, the sample sizes were pretty small. So my competitive edge sample was 14 students, and then my URG group was 11 students, and then my well-represented group was over 50 students. And so, of course, that's sort of limiting. And like you sort of got at some of the difficulties of social science of, finding these control groups. And then also, just there's also this bias of who chooses to respond to your surveys. And like we know, often the people who respond to surveys are the people who have the most extreme opinions. We often lose people sort of in the middle. I did also use like a gift card raffle as some incentives, and like also did a lot of work with the competitive edge staff to help them also encourage students to fill out the survey. But yeah, to sort of summarize that, I did find with these 2 control groups I looked at, I did have a pattern where the competitive edge students seemed to improve more in 7 of these 8 skills. You know, 2 of these differences were statistically significant. And I can, you know, did like a power analysis and looked at, I would have to have a really large effect size or really big difference in these students' changes in skills to accurately detect it, looking at the competitive edge minus the URG students. Like this 1.5 is considered a large effect size. Yeah, but so yes, I did think about and look at some control groups, but sort of left it out in the summary of my talk. Thanks for the question, Victoria, and letting me pull out the, you know, everybody's dream of like, I have slides ready for this. So thank you for that. Oh, somebody asked here, what is WR? It's well represented. So the students who are not historically excluded or underrepresented. Oh, and then somebody asked about let me find, you know, we're running up on time, so lots of people have to run. The DFE mean that wolves have a higher likelihood of having traits that are deleterious. Yes, that's true. It is. And this is something Eduardo, who did a postdoc in Kirk's lab, is thinking more about and working on a paper on, of just sort of describing and thinking about what does this mean that wolves have this surprisingly strong deleterious variation. Because you know, working with people in Kirk's lab who are used to looking at these plots, when I have shown them my wolf plot, often there's a little bit of squinting and like, are you sure that's right? Like, what's happening there? But me and Eduardo and Meishi and other people, we've all sort of looked at this and are like, yeah, that this seems to be the estimate, where wolves are biased towards this strongly deleterious variation. All right. Any other questions?
Let's congratulate Tina.